those. All right, we're going to go through our lab on mitosis, meiosis, and gametogenesis. So if you could look in your lab supplement and turn to this page. It's page 1 in your lab supplement, has the objectives for today's lab. So I'm going to keep coming back to this throughout class just to make sure we're covering everything. So the first objective has us just looking at some terminology: somatic cell, gamete, zygote, haploid, diploid. So in the reading, those terms are defined pretty simply here for you. Somatic cells are all body cells. So the key word for somatic is body cells. So every cell in your body outside of the sex cells, sperm and egg, we call somatic cells. So a majority of the cells that make you are somatic cells. They contain 23 different chromosomes, and you have a pair of each of those. One set of 23 you got from your mom, and the other 23 in all of your body cells you got from your dad. So you're a combination of the chromosomes from your mom and dad, but each 23 for a total of 46. So there's 23 different human chromosomes. We inherit one of each from our parents. So we call that chromosome number 2N. N is the number of different chromosomes we have in the body. And how many did I say we have? 23 different chromosomes. And we have 2 pairs of those. So we say the somatic cells are 2N. We have 2 pairs of 23 chromosomes. Or 1 pair, I guess. I mean, if you want to be technical, 1 pair of each, right? So 23 from our mom, 23 from our dad. And then the gametes are the sex cells. So it's another name for sex cells. So sperm and egg are gametes, and they each have 23 chromosomes. And that's important because when sperm and egg come together during fertilization and the nuclei of the sperm and egg fuse, 23 plus 23 is 46, and that makes a somatic cell. So that's the beginning of you, and that very first cell formed from sperm and egg, the nucleus of each uniting to get 46 chromosomes, is called a zygote. So a zygote is a one-cell organism. That's how we start. That's the very first step when those 2 nuclei fuse with sperm and egg. So how many chromosomes are in the zygote? Did I just say? Sperm and egg fuse, each bringing their chromosomes together, 46. So diploid means 2 chromosomes of the same kind. So we have 23 different chromosomes. So we have 2 sets, 46. That means we're diploid. And if you have 1 set of chromosomes, we say that cell is haploid. So that's just some terminology to kind of get things going here. Now I have a PowerPoint also for the first part of today's lab. This is the mitosis, meiosis portion. So to review mitosis, a couple of things to remember. This is from General A&P. Mitosis is just making a copy of a cell. So anytime we need to replace or repair or add another cell, we undergo mitosis. So what are some times in our lives from beginning to end that we might need to do those things? Replace, repair, add new cells. Yeah. Yeah, our skin sloughs off. We need to replace more, so that occurs in the bottom layer of our epidermis, the stratum basale of the epidermis undergoes mitosis. Cardiac muscle cells, do they replace themselves? Not necessarily. If they die, they're gone. But during periods of growth, as the heart starts from the size of a baby to a size of an adult, we would replace heart muscle cells in that case. There are some tissues that can grow back, like the liver can regenerate. If you lose a part of your liver in, say, an auto accident, you have lacerations on the liver, that can repair and make new cells to replace the damaged ones. Bone cells definitely replace themselves. If you fracture a bone, you can repair that. Your body will repair that by mitosis. So those are examples of replacing. And during periods of growth, anywhere from infancy to end of puberty, that's all mitosis, occurring constantly and making new cells. So again, when we look at mitosis, it's just making a copy machine. The cell that we're copying is the same.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes.
So in mitosis, before we kick into mitosis, our cells are hanging out in interphase, where you have a nuclear membrane, you have this kind of loosely held DNA called chromatin, and all the organelles of the cell are just kind of doing their thing, undergoing metabolism. But when it comes time to replace or repair a cell, we kick into mitosis, and all those organelles disappear, except for those that are important for cell division, like the centrioles. And the nuclear membrane breaks down. So this is the first stage of mitosis. When we're getting ready to divide, the chromosomes start to coil and form, or the DNA starts to coil and form chromosomes, those X-like structures. So those are, these X-like structures are called chromosomes, and these are strands of DNA that have been replicated. So remember last week when you had to replicate the DNA, right? We gave you one side, and then we had to make a copy of DNA, essentially, right? So this is replicated DNA. So this is one chromosome that's been copied to make another chromosome, and then it's attached by a centromere. So this is one chromosome that's been copied or replicated. So if I count the number of chromosomes in this cell, I have 1, 2, 3, 4, 5, 6 chromosomes in this cell. Each has been copied, but we count it as one chromosome when they're tethered together by that centromere. Remember the centromere that we learned in general A&P? That's what attaches those two sister chromatids together, those two pieces of DNA. So we have 6 chromosomes here, and 3 are blue and 3 are red. So we have one set from mom, say the reddish color ones came from mom, and the other set came from dad. So that's a total of 6 chromosomes, 3 different chromosomes, right? We have a pair of each type.
Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates.
So this is prophase again. Again, we know nuclear membrane, centrioles are on either side. We can see chromosomes in the middle, but they're not quite lined up in the middle. They're just kind of hanging out. And this spindle is being secreted by these centrioles, and the spindle is going to attach to the chromosomes and push them to the middle. That's the next phase of mitosis. And what is that called? Metaphase. Meta means middle, so they're lined up in the middle.
With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles.
So this is the next phase. When the chromosomes are being pulled apart, what is that phase? Anaphase, being pulled apart. So ana, apart. So for the lab exam, you're going to have to look at pictures or slides of the different phases of mitosis and be able to identify it. So it's a little bit of a review from general A&P. Not many questions, but a couple.
Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating 2 new cells in this ongoing cycle that sustains all life.
Now see how that cytoplasm is pinching inward? This animation, it happens a little later in the animation, actually happens toward the end of anaphase is when that pinching in occurs. So the last phase of mitosis where the pinching in occurs but the nuclear membranes haven't appeared yet would be called telophase. So look for that figure 8 appearance for telophase in the microscope. And then you'll see the dark chromosomes in either side of the cell. And this process of the cytoplasm pinching inward is called cytokinesis. That's another term that you should be aware of. So the pinching in of the cytoplasm is called cytokinesis. So we have 2 new cells. So going back, chromosome number stays the same. We had 6 chromosomes at the end of mitosis. We still have 6 chromosomes. But are they as sister chromatids anymore? Do they look like X-like structures? No, now they're single-stranded, not stranded, but 1/2 of each. It's still double-stranded DNA, but we only have 1/2 of that X-shaped structure that we started out with. So the chromosome number stays the same. Now the other process is much more detailed, but it's still very similar to mitosis, and that's meiosis. So where does it occur? Testes and ovaries, that's it. So 2 locations for meiosis, testes and ovaries. And it forms gametes. So the cells at the end of meiosis are sperm and egg. So the role of meiosis is to cut that chromosome number in half. Another name for meiosis is the reduction division. So it's the division of cells that reduces the chromosome number by 1/2. So there's a meiosis 1 phase and a meiosis 2 phase. And at the end of that, if you think of meiosis kind of like mitosis times 2, instead of getting 2 cells at the end like we do in mitosis, at the end of meiosis we get 4 cells. So it's almost like mitosis runs twice. But there's some slight differences in those processes, which we'll look at.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring.
And what do we call that single cell that's diploid when sperm and egg fuse? Zygote. Yep, good.
Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA.
So
Meiosis involves 2...
So there's my chromosomes that have doubled, but they're still considered 1 chromosome. So we have a total of 2 chromosomes starting out in this cell, the green chromosome and the purple chromosome. And where did each of those come from? Mom and dad. Okay. So they replicated. That's what makes them look like an X. As DNA replication happened, we have another copy of that DNA attached to that parent copy. So now we're going to undergo meiosis to get this chromosome. So there's, in this example, this would be 1 type of chromosome, and we have a pair of them, right? So there's 2 chromosomes here.
2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells.
So this meiosis 1 is going to take these 2 chromosomes and go through prophase 1, metaphase 1, anaphase 1, telophase 1, just like we saw in mitosis, same look, and we end up with 2 cells at the end, just like with mitosis. Then this cell is going to undergo meiosis 2 and undergo prophase 2, metaphase 2, anaphase 2, telophase 2, just like mitosis, to get these 2 cells. And the same thing's going to happen with this one. It's going to go through meiosis 2 and end up with these 2 cells. So it's just like mitosis, except for there's some slight differences.
Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of 1 chromosome are no longer identical to one another.
So this process is called crossing over. But 2 other terms you need to know if you look at your worksheet for today. One is, let's see, lab handout. Oop, not that one. Where's the worksheet? Here. Genetic variability happens during meiosis. So what leads to that? The first one is, it's called synapsis, is when those two chromosomes came together and it formed that four-armed structure where the two X's came together. That's called synapsis. And that structure is called a tetrad. Tetra means four. So if I go back to my animation here, so this would be synapsis is one thing that leads to genetic variability. And that's what happened here, is when you saw those four together. That process right there is called synapsis. It's just the coming together of like chromosomes. So we have 23 different chromosomes, so this could be chromosome number 1. One from mom, one from dad. It's the same chromosome, carry the same type of information. We call those homologous chromosomes. Two chromosomes that carry the same type of information. Different versions of that information, because your mom and dad don't look the same, but they are humans, so they have those 23 chromosomes with similar information. So they came together, that's called synapsis. That structure is called a tetrad, because you can see the four strands are part of a tetrad. Then when they exchanged that information, again, that was called crossing over. So the arms of the homologous chromosomes overlap and they swap DNA, just little pieces of DNA. So it's not completely different, but this chromosome now has a little bit of mom on it, and this chromosome has a little bit of dad on it. They both take place at prophase 1, exactly. So crossing over is the second one, and that is also prophase 1. And the last one is the next stage in the process.
Adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random.
So what that means is this chromosome could have went on this side of the cell, and this chromosome could have went on this side. So how they line up on the middle of the cell is random. So when this cell divides, like this one here is mostly mom, right? So this cell, when these two cells divides, this half is going to have mostly mom's characteristics on that chromosome, right? But if they randomly went the other way, the green one went on this side, then this cell would have mostly dad's characteristics on it. So if this happens to be the sperm that makes baby, that child's going to look more like dad because it inherited that chromosome that had mostly dad's DNA on it. And you've seen that in families, haven't you? It's like, oh, I can tell whose baby he is, right, or she is, because they look so much like the parent. Or sometimes you have a kid that looks nothing like either parent because there's so much variability that happened in this process. But the goal is variability in meiosis. So this process of independent assortment is what we call this, or random assortment. We call that happens during what phase? Yeah, metaphase 1. So independent or random assortment, either term is acceptable. That's the third one for letter C, and the stage is metaphase 1. Now there's another link to variability that's not on the worksheet, and that is which sperm is going to fertilize the egg. There's millions of sperm in one ejaculate from a male during orgasm. So which one is going to fertilize? That's random also. So we call it random fertilization is another source. That could be letter D. It's not on the sheet. So you don't need to know it for the lab exam, but it's important to know that that's another reason for that for variation. So that's why when you see a woman who has twins, one's a boy, one's a girl, or even if they're two girls and they look nothing like one another, that's because they're fraternal twins, which it was two eggs that were ovulated by the mother and two different sperm that fertilized each of those eggs. So they're no more related than brothers and sisters born 5 years apart. They were just shared a womb together. Okay, so back to meiosis.
Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell.
So we still have this X form, the homologous chromosomes, but they were separated now. This chromosome was separated from the other one just like it on the other side. So there's 2 chromosomes in this new cell. Would you agree with that? Right? If I go back to the beginning, when I started out, there were how many chromosomes? 4. So meiosis 1 takes the cell from 46 down to 23. So at the end of meiosis 1, we have 2 cells, each with 23 chromosomes. All right, so now we'll continue.
After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber. And in telophase 2, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Okay, so 4 different cells. Has the chromosome number changed from meiosis 1 to meiosis 2? Yes, no? Louder? No. We had 23 at the end of meiosis 1, and we still have 23. But what's the difference when looking at mitosis or meiosis 1 and meiosis 2? I made a little worksheet here for you to help with that. It has a model. Let's go to this here. It's called Models and Slides document. Be sure that you look at this and label it or understand the different parts. So here's the model we have in lab of mitosis. So what would this phase be here? Say it louder. Interphase, right? Because we can see a nucleolus, see a nuclear membrane. Number 2 is 2 and 3 are prophase. 4 is metaphase. 5 is metaphase. 6 is anaphase. They're being pulled apart. And 7 would be telophase. Cytokinesis is occurring, and at the end, we're done. So that's mitosis. Now, if I look at this model here, I can make it a little bigger. Okay, so if I look at this model, this is our meiosis model. So here I have interphase, right? Now I see some chromosomes showing up, so this would be prophase 1. This would be prophase 1. Here I have my chromosomes coming together. What do we call this process where they come together? synapsis, and this structure, this four-stranded structure, tetrad. So I have two homologous chromosomes that have come together, synapsis. They're swapping some DNA. What do we call that? Crossing over. So this is prophase 1. Look for the two X's next to one another, because we can take each of these little pieces out of here and put these on as a separate question on the test. So don't memorize these in order. I mean, it helps to learn it that way in the beginning, but be able to pull them out separately. And a really good idea would be to print this off and make flashcards and then put it on the other side what phase it is. Okay, so they're in order for you now, but cut them up, make flashcards, and it'd be a great way to study. So this is prophase 1. This would be what comes after. They're lined up in the middle. Metaphase 1, anaphase 1, they're being pulled apart. Telophase 1. Then this would be anaphase 2. They skipped over prophase 2 and metaphase 2 and went right to anaphase 2. And then this would be the end, telophase 2. So now, here's a little hint. If you look at the model, if it has the chromosomes in the version of an X, which means you have sister chromatids, you can see the chromosomes has that X structure, then you know you're in meiosis 1. So if you can just identify, are they lined up in the middle, are they being pulled apart, do I see X's, you're in meiosis 1. So you're going to say prophase 1, metaphase 1, anaphase 1. If they're as single chromatids, see they're not X's anymore in these examples, then you know you're in meiosis 2. So it's going to be, this one to be anaphase 2. So look for the single strand, you know, the single sister chromatids being pulled apart in meiosis 2. Does that make sense? All right, going back to our objectives then. So we talked about mitosis. We looked at the different phases of meiosis. We talked about the different processes and when they occur. When does chromosome reduction occur? The specific stage? Telophase 1. Very good. Telophase 1. Crossing over, we said, occurred prophase 1. Metaphase 1, independent assortment, and synapsis tetrad was prophase 1 also. So comparing the two, let's go back to the PowerPoint then. If we compare the two processes, and these are just the processes described, so you can watch that again if you want. But if I compare the two processes, mitosis and meiosis, we both start out with diploid cells. Meiosis is going to multiply that cell. We'll talk about this when we get to gametogenesis. It'll make more sense. But let's just start here. So we're going to have this cell that's going to divide into two haploid cells. Mitosis forms two diploid cells that are the same as the parent cell. This cell is going to divide again during meiosis 2 to form two more haploid cells. Same thing with this one, two more haploid cells. So comparing the two, 4 haploid cells at the end versus 2 diploid cells at the end. Everybody agree with that? And then we have some variation, you know, genetic variation that happens in prophase 1, metaphase 1, and telophase 1. Here's just a written description of those two processes. You can read that on your own.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
So here we have 2 cells, 2 chromosomes. Here you have 4 cells with half the number of chromosomes. They both started out the same. Okay, so the next slide, PowerPoint, is looking at gametogenesis. So now that we understand what mitosis and meiosis is, now we're going to apply these processes to the process of making sperm. So spermatogenesis is the process involved in making sperm cells. Oogenesis, O, 2 O's together, we say in science oogenesis is how you would say that, is the process of making eggs in the female by meiosis. And gametogenesis is the 2 combined. So the process of making sperm and egg through meiosis, we call that gametogenesis, making the gametes. So spermatogenesis, making sperm, oogenesis, making egg, gametogenesis, either one. Spermiogenesis refers to the process of making a viable sperm. So you have a haploid spermatocyte, or spermatid, and we want to turn it into a spermatozoa. So it's just the process of sperm development and maturation. So that's just when we already have the sperm, we're just developing it to become a viable swimming one-celled structure. So that's spermiogenesis. It's just sperm development, the kind of the late process that occurs. So we're going to start with spermatogenesis and then end up with gametogenesis. I'll go back to our Blackboard page. Here we go. I have a handout here. It's a blank diagram. I'm just going to pull this up. This is for you to label and practice with. This is spermatogenesis here. This is oogenesis. What I would recommend is that you name these cells and give them a chromosome number of either N or 2N. And that's going to help you on the test because he'll have models, diagrams, written questions in the lab exam related to the names and the diploid number or chromosome number of these cells. So this is what's going on in the ovaries. This is what's going on in the testes. And these are both from those diagrams are both from your textbook. So I'll just keep that hidden for now. Keep that hidden. And here's the PowerPoint. Okay, so gametogenesis, process of making gametes. So these come from the starting cell that leads to sperm and egg. We call those germ cells. So they are destined to be sperm or egg. So somatic cells are body cells that are just part of the embryo. They're growing and making more of a tissue and making that tissue bigger and bigger. But germ cells are destined, they're like stem cells, right? You've heard of stem cells. So these are unspecialized cells, but they're specialized to the point that they're going to become sperm and egg. So they're not completely blank. They are destined to be sperm and egg, but they're very early cells that have not differentiated. So if you have a mutation in a germ cell that was used to form you, like your dad when he was forming sperm cells, there was a mutation there in that process, then that would be inherited. You would inherit that change in your DNA. And that would be a germline mutation, like we said, is inherited. Somatic mutations, remember we said, can cause cancer. They can happen in any body cell. Just they're isolated, where germ cells would be in every cell because you inherit that DNA from the very get-go as a zygote. So let's talk about spermatogenesis. So sperm are made in the testes, and the specific structures within the testes where the sperm are formed are called the seminiferous tubules. So if you look at the testes, do a cross-section, and your textbook has a nice picture of what the seminiferous tubules look like from the outside. So if you cut a testes in half, they kind of look like, if you know what ramen noodles look like, it's just a network of tiny tubules. And what we're looking at in this diagram is a cross-section of one of those seminiferous tubules. And can you see the hair-like flagella at the end of a sperm that are kind of clustered toward the center of that picture? So that's the center of the seminiferous tubule. And the wall of that tubule is where those sperm cells are formed. So out here at the very edge is where you would find the germ cells that are diploid, that are destined to become sperm. So as sperm develop, they work their way inward inside this tubule until they break free and with their tails swim away. And then they enter the vas deferens and out and enter the female and fertilize the egg. So here's a tissue slide of a seminiferous tubule in the testes. So the organ is the testes, the specific structure in the testes is the seminiferous tubule. And you'll need to know all of these structures other than the sustentacular cell. You don't need to know that one, or the basement membrane, but you do need to know all these other cells in a tissue slide for the lab exam. So on the very outer edge, I said, are the very beginning cells that are diploid. So we'll pick this one here that has the label. Spermatogonium. More than one spermatogonia. So a spermatogonium, the way I remember this, it sounds kind of silly, but it's going to be a sperm, okay? Using farm language, Midwestern slang. Okay, so it's destined to be that, but it's not there yet. So this is a diploid cell. It has 46 chromosomes, or 2N. It's on the outside, destined to come and become a sperm cell. So 46 chromosomes, and this diagram I took from, or the next diagram I'm going to show here is a really good explanation is figure 27.9 in your textbook. So let's go to this picture. This is the one from your textbook. 27.9, figure 27.9. In the newest edition, it's on page 1040. And that's the one that I have attached in Blackboard as a blank copy for you to label for practicing. Okay, so what happens here is we have this stem cell, and this stem cell out here on the edge is, again, a spermatogonium, and it undergoes mitosis to form another cell that's going to move on and become a sperm. So this one is just, you can still call it a spermatogonium. They call it a daughter cell. It's from mitosis of this cell. So if this cell undergoes mitosis, wouldn't I have two? I'm saying mitosis, so that means two cells that are each 46. Would everybody agree with that? Right? So this one is going to stay. The type A daughter cell stays here to keep that DNA for future sperm. And then this one is going to come down and divide and leave the edge of that seminiferous tubule. So this one's going to come down. It's going to divide by meiosis 1 into a primary spermatocyte. And a primary spermatocyte is 2N. Undergoes meiosis 1 to form secondary spermatocytes. So secondary spermatocytes, if they underwent meiosis 1, what would the number here be for chromosome? Haploid or diploid? Haploid. So 23 chromosomes in each of these. So the secondary spermatocyte has 23 chromosomes, or haploid. The primary spermatocyte had 46, or diploid. Then this cell is going to divide into two to form these two cells. So this one divides by meiosis 2 to form these two. This one divides through meiosis 2 to form these two. So we have a total of 4 after meiosis 2, just like we saw in the generic example we went through. And these are called spermatids. So how many chromosomes in the spermatids? 23, or haploid again, right? So you write the letter N there. So you can label that in there. These are all N. And their names, early spermatids. These are also N. There's only 2 of them, secondary spermatocytes, and this is 2N, 46 chromosomes, primary spermatocyte. And then out here on the very edge, spermatogonium, also 2N. So then this process of taking these spermatids and forming the cytoplasmic bridge, condensing the material, the intracellular material, into a neck and a flagellum, so they're active swimming sperm, then we call them spermatozoa. But that whole process going from spermatid to spermatozoa That process is called spermiogenesis. Spermiogenesis. So when does this process occur? When we go from spermatogonia to spermatocytes, so we're starting sperm development, it starts at puberty. So boys don't start making sperm until 12, 13, 14, all the way up until death. It slows down for sure because there's other cells that influence sperm development, and that's the cells that secrete testosterone. So these cells in between the seminiferous tubules are called interstitial cells. Inter means between. So they're between the seminiferous tubules, these little clusters called interstitial cells. Their job is to secrete testosterone. So these cells become a little sluggish as men age, and they don't secrete as much testosterone, which doesn't support as much sperm development. So sperm development slows down with age. But it still occurs. And if a 90-year-old man is able to participate in intercourse, he could, father a child. Or they could even go and retrieve the sperm from the epididymis and take sperm that way and, do it through in vitro fertilization, and that has happened. So it begins at puberty. Talked about what is formed in meiosis 1. It forms 2 secondary spermatocytes. At the end of mitosis 2, we have 4 spermatids. Again, they're not moving cells. They're non-motile, but they're close to the inside edge. So they're close to that hollow center, ready to fly, ready to swim, but they have to undergo spermiogenesis to have the flagellum, the head, and the neck, and proceed. So that's the different parts. So spermiogenesis is that process of spermatid to sperm. So how long does it take from becoming a spermatid to being a mobile sperm? About 24 days. So here is a diagram nicely labeled with all the different chromosome numbers. So this is a great study tool. It has the name of all the different cells. It shows you the chromosome number along the way, and just know that it works from the edge of the tubule inward. So let's watch this animation. This is a really good animation that illustrates the process.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
And they're often swimming. All right, so let's look at the worksheet. Do I still have that pulled up somewhere? No, I don't. All right, so we'll go back to this and look at so number 11, in what organ does spermatogenesis occur? The organ, testes, and the structure? Seminiferous tubules, good. And then we'll go through the rest of these after we finish up oogenesis. Okay, one other thing I want to refer you to, and that is the worksheet that has the different structures on it, this one. So we looked at those. Here's a mitosis slide. So if you look at this slide, which phase of mitosis is this representing? Anaphase, and this one? They're kind of lined up along the middle. Metaphase, this one also. Metaphase, how about this one here? Cytokinesis, yeah, at the end of which phase? telophase, because we're at the end. You can see they're almost separated, but not quite, and you can see visible chromosomes, no nuclear membrane yet, so that's telophase. And each of these with just a regular circle and the visible nucleoli, the nucleolus in the middle of that nucleus, these are all interphase. So if you don't see any chromosomes and you just see a pink or dark circle in the center, that's a nuclear membrane, and that's just a resting phase, interphase. So I put this up here as kind of something to refresh your memory on. So here's a slide of a seminiferous tubule. So out on the edge here, these cells would be it's on the very edge. It's going to be a sperm stuck in the edge. Spermatogonium. So if it's right here on the very edge, if you see a cell pointer on the edge, spermatogonium. How many chromosomes? 46. If I point to these cells here that don't have tails yet, but they're here, what would these be? Spermatids. How many chromosomes, or what's the description for chromosome number? 23, haploid, or N. Okay. And then if I go in the middle here, these might be secondary spermatocytes, but it's really hard to see if they're, secondary or primary. But we can, with reasonable degree of confidence, say that these are the spermatogonium that are on the edge. These are the spermatids here on the inside lining, and if they have a tail attached, then they would be spermatozoa, right, at the very end of that development. And what do we call these cells in between the seminiferous tubules here? Say it louder. Interstitial cells, yes, interstitial cells. So here's another one that's been labeled for you, which is nice. So you can look at that, use that for a study tool. So this is all in that handout I attached. Okay, so now let's compare this process to what goes on in the ovaries. So in the ovaries, not on the edge, the ovaries are just a mass of tissue. So they're just a mass of cells and tissues, different types of cells, about the size of a plum, maybe a little smaller than that. Maybe a large grape is about the size of what an ovary is in the human. So there's these cells that are germline cells, just like we saw in the spermatogenesis, but in the egg, or in the ovary, they're called oogonia, or one of them we call oogonium. It undergoes mitosis. One is going to stay in oogonium, because we don't want to lose that DNA. So one is going to stay in oogonium, and one is going to grow and develop and become the different cells of oogenesis. So an oogonium that grows and develops becomes a primary oocyte. So just like we had the primary spermatocyte that had 46 chromosomes, the ovary has a primary oocyte with 46 chromosomes. So it undergoes meiosis 1 to form 2 secondary oocytes, but we don't get 2 secondary oocytes like we would expect. What we get, how many eggs do we need? What is the human body designed to grow and develop? How many babies? 1. That's ideal, right? For our species, 1 child is enough, because how long does it take before that child is mature and ready to leave home? 6 years? No. But it's pretty helpless, right, when it's first born. It's not like a pony that comes out and is running across the pasture hours after birth, right? Maybe not running, but wobbling across the pasture. Newborns can't do that. So we're designed to have 1 baby. So we only need, women, we don't need 1 million eggs, right? Men need 1 million sperm, because a lot of those sperm aren't going to make it up the reproductive tract to find that egg. So more volume, or higher numbers, I should say, of sperm are going to ensure fertilization. But women, females only need 1 egg to be fertilized. So only 1 egg is actually developed in this process of oogenesis. So we have the oogonium, and at the end we're going to have 1 healthy, mature egg after meiosis 1 and meiosis 2. So here's what happens. Just like in spermatogenesis, this primary oocyte splits, But instead of having two secondary oocytes, we have a polar body. Look at how much smaller it is. Most of the cytoplasm and the contents of this original cell went to this secondary oocyte because this is the one that we want to become a mature egg. This one is a polar body. It's just kind of fizzles away, doesn't have enough information to become an active living cell. So we call it a polar body, okay? So that's meiosis 1. Meiosis 2, what happens? Meiosis 2 occurs, and we have a mature ovum, and this might, and another polar body, right? Because this is going to split into two after meiosis 2, so we have an ovum and another polar body. And this polar body, if it has enough information, it could undergo meiosis 2 again. And we should have 4 at the end, right? Just like we said with meiosis, we always have 4 haploid cells at the end. So if this one did divide again in meiosis 2, we could have 1, 2, 3, 4 cells like meiosis 2 should provide. But with eggs, we really only need 1 mature egg. So these polar bodies degenerate and they don't really have any function. So this is where all the action is. This is what we want. So this is a haploid cell. So oogonia becoming primary oocytes, that's just mitosis. We just make a copy of that cell. They're both 46. They're both diploid cells. When do these form? Before that woman is even born is when all these primary oocytes are formed. So this mitotic process of taking oogonium and making a primary oocyte happens before birth. 7 million primary oocytes. That'd be a lot of babies, wouldn't it? So we don't need that many in our lifetime. A woman only produces 1 egg a month during her reproductive years to be fertilized. So that number is cut down throughout life. So only 1 million of the 7 million produced will remain at birth. Some of them degenerate. They undergo apoptosis. And then from birth to puberty, only 300,000 remain. That's still a lot of kids in a woman's lifetime. So she's never going to use all 300,000 of those eggs. So what happens then is the primary oocytes enter meiosis 1, but they're stuck in prophase 1, metaphase 1 until puberty. So this development, this meiosis is occurring during childhood, but it stops until puberty. So it's stuck in that prophase 1, metaphase 1 phase. So here's an ovary. So on the very edge are these primary oocytes, and the cells around them are called follicle cells. They just nourish that nucleus, that developing oocyte. So they're just supportive cells. And they're much smaller, sometimes found near the edge, but not always. And then when it comes time for puberty and ovulation leading to fertilization, some of those cells will be picked to develop and become a mature oocyte, a secondary oocyte to be ovulated. And that's what this one is. This is a secondary oocyte that was ovulated, released from the ovary into the fallopian tube, waiting to be fertilized by a sperm. So the process of going from primary oocyte to secondary oocyte then is each month after puberty. During the menstrual and ovulatory cycle is when this development from primary oocyte, meiosis, so which stage of meiosis are we in? 2N to N? Meiosis 1 or meiosis 2, or both? Both, yeah. Meiosis 1 is going to cut the chromosome number in half, right? So the primary oocyte to the secondary oocyte is meiosis 1. Then meiosis 2 begins, but that secondary oocyte is stuck at metaphase 2. So when this secondary oocyte is released at ovulation, it's stuck in metaphase 2. So the chromosomes are lined up on the center of the cell. It's ready to separate, right? But that has not happened yet until fertilization. If fertilization occurs, then it splits and metaphase 2 completes, and meiosis 2 completes only after fertilization. So it's stuck in metaphase 2 until fertilization. So some questions you might see in the lab exam is when these cells are formed. Okay? So a primary oocyte is formed when? A primary oocyte is just mitosis from a oogonium. So after birth, let's go back, primary oocytes are formed during the fetal period, and then their number is just reduced up until puberty. So secondary oocytes are not formed until after puberty. So primary oocytes are formed during the fetal period. So again, this is a nice chart because it tells you when everything is occurring. So before birth, childhood, puberty to menopause. So these are the reproductive years here. So here we can see, again, mitosis takes that oogonium, forms a primary oocyte. And that primary oocyte is going to just hang out until puberty. Then it will be chosen to enter meiosis and become a secondary oocyte, and it'll be stuck there until penetrated by a sperm. When it's penetrated by a sperm, then it becomes a mature ovum. So after fertilization, we have an ovum. So this is, again, 4 cells. We know that meiosis produces 4 haploid cells. But again, 3 of those cells are polar bodies that are not useful, not necessary for reproduction. The egg is what we care about, the ovum. Now there's another column of information here, and we're going to go back and talk about that. So these cells, like I said, the supportive cells around the developing nucleus and cytoplasm of this oocyte are called follicular cells, follicle cells. So when the follicle bursts and releases that secondary oocyte, some people feel pain at ovulation. And when I taught health years ago at UWL, sometimes we'd talk about fertility and reproductive system. And I had a student come up and say, I'm worried that I might have ovarian cancer or some problem because I get really bad pains every month. It lasts for a couple of days, and I can't hardly sit down. I'm in such pain. I have to take ibuprofen, and it's not when I have my period. I'm not having my period when I have these pains, and it's really bad sometimes. And what she was experiencing was ovulation pain, is when that ovary ruptures and releases that secondary oocyte, prostaglandins are released, and that causes pain over that side. So if you're female, you may have noticed, if you notice, some people can't feel ovulation, some can. You may have noticed you have a dull, aching pain about 2 weeks before the menstrual period would start, and it alternates. Now, the ovaries don't always go left, right, evenly. Sometimes might be left, right, left, right. There's no real pattern to it, but we know that the ovaries both, take turns. Only one ovary releases 1 egg each month. But when you get to be over 35, there seems to be a peak between the age of 35 and 40, where there's a period of time where women tend to release 2 eggs, and that's where you see an increase in fraternal twins, because they release 2 eggs that particular month. Does anybody know anybody who had twins? They got pregnant over 35? You know somebody? Yeah. And they were not identical, correct?
Actually, not quite sure. I don't know it that well. I just had them.
Oh, okay. So it's hard to tell. All newborns look the same, right? Yeah. But typically they're not, because it was 2 eggs, 2 different sperms, so 2 different babies. But anyway, so these follicle cells, so then that follicle is called a corpus luteum. Luteal refers to the color yellow. It's like a Latin or Greek prefix, I'm not sure which one. But so this is a special, organ within, it's a tissue, I should say, within the ovary that starts secreting hormones, and it secretes progesterone. And if this egg became fertilized by sperm and pregnancy start, that corpus luteum is going to continue to secrete progesterone, which supports the uterus and maintains a pregnancy. So that's a really important structure, and it's the remainder of those follicle cells after the egg has left, after the secondary oocyte has left. And some women will find when they are early pregnant that they have a little discomfort on the side, even though they're pregnant and baby's in the uterus, you know, down low between the hips. They feel that ache on that one side. And again, that's a corpus luteal cyst that can remain there, that was the egg that was there from when baby, or when that secondary oocyte was ovulated that formed baby. Some people, the cyst gets quite large and can be damaging or dangerous. I know someone who, they talk about maybe having surgery to remove that cyst. I don't know. I don't think she ever had to have surgery, but it was a concern for a while. So I don't know what happened there, but that's the corpus luteum. But now if pregnancy did not, and it'll grow bigger until the placenta grows. And the placenta is what attaches to baby, and we'll be talking about that next, 2 weeks when we talk about reproduction and fetal development. Then the placenta will take over all that hormone production, and the corpus luteum will shrink down and become less important. So this is just comparing, well, one more thing I should talk about is, there's an animation I'm going to go to that talks about the corpus luteum before we go any further. Question?
Yeah, so do you know if after someone was pregnant and they had to take like progesterone like supplements, that like the corpus luteum wasn't doing its job?
Exactly. Yep. And that happened with one of my pregnancies. It's just one of those weird things. And it was not my last one. It wasn't an early one. It was just one of those weird things. And it's a good thing to know that, because some women who have a lot of early pregnancy losses, early bleeding and loss of the pregnancy after a positive test, it can be something as simple as low progesterone. And taking getting a blood test, checking progesterone levels, they can supplement a woman with progesterone until that placenta kicks in, and then the placenta does the job and they don't have to supplement with progesterone anymore. And it can prevent a loss of a pregnancy. So really important test. Okay, so let's watch this corpus luteum. And if pregnancy doesn't take place, secondary oocyte here is not fertilized, then this breaks down and becomes a corpus albicans. So it's a white structure that we see on the ovary. And what do we call a person that has white skin, white hair, and red eyes? Albino. Alba means white. So that's where corpus albicans comes from. That'll help you remember that word. Alba meaning white. Luteal means yellow.
A primordial follicle consists of an oocyte surrounded by a single layer of squamous granulosa cells. A primordial follicle becomes a primary follicle as the granulosa cells become enlarged and cuboidal. The primary follicle enlarges, and the granulosa cells form more than one layer. The zona pellucida forms around the oocyte. The structure is called a secondary follicle when fluid-filled vesicles develop among the granulosa cells and a well-developed capsule, or theca, becomes apparent around the granulosa cells.
And there's a lot of extra terminology in this video that you don't have to know. You don't have to know all the different names of the follicle cells. Just know that this is the, what is this? The oocyte. And then we're going to talk about some of the structures after it's ovulated.
The theca possesses internal and external layers. The structure is called a mature follicle when the fluid-filled vesicles form a single antrum. At full maturity, the oocyte is located in the cumulus mass. During ovulation, the oocyte is released from the follicle, along with some surrounding granulosa cells of the cumulus mass, called the corona radiata. Following ovulation, the granulosa cells divide rapidly and enlarge to form the yellow corpus luteum. When the corpus luteum degenerates, it forms the white corpus albicans.
Okay, so when this secondary oocyte is ovulated, there's some follicle cells that remain around it. And this is a term that you do need to know, and that's, those cells are protective cells called the corona radiata. And the sperm have to penetrate through those cells to get to that egg. And there's another layer, it's called the glycoprotein, so it's a protein with sugar attached to it, called the zona pellucida. See the white here? It's smooth, it doesn't look cellular, it's just a smooth outer covering of that egg, again called the zona pellucida. that the acrosome, if you remember the enzyme in the head of the sperm from general A&P, that has to dissolve that zona pellucida, cause a chemical reaction to allow that sperm nucleus to enter that egg. Once that sperm enters the egg, the zona pellucida hardens, almost like an eggshell, you can just use that analogy, so no other sperm will enter. Because there's millions of sperm working their way through the cells of the corona radiata trying to get to that egg. But once the first sperm gets there, that zona pellucida hardens and shuts down and will not allow another sperm to enter. Because we don't want more than one sperm, because that means more chromosomes, and we have to have 46 chromosomes to turn into a viable zygote human being. Okay, so here is the zona pellucida, that white outer layer. So you will see a slide in the lab exam about the zona pellucida, so be sure that you are able to identify that. And then the corona radiata would, again, be these cells around the edge here. And in that other document here, I have a picture of that. This is the only one I could find that had the corona radiata, or yeah, the corona radiata, all those cells, corona radiata, and then this white, even layer, zona pellucida. Here's an ovary with a large corpus luteum. So these, can you see these empty follicles here? But this is the large corpus luteum that actually ovulated that secondary oocyte. So that's what this is a picture of. So be sure that you look at this handout here and identify the structures. So again, primordial follicle. So we see, one cell, not a lot of follicles around it. Here's a primary oocyte, getting a little larger here. Secondary oocyte is much larger, has a big fluid-filled space. I'll have to put another picture. I don't see that I have one of a secondary follicle. I'll find one and put it up on Blackboard. Okay, so going back then, comparing the two processes, spermatogenesis and oogenesis. Spermatogenesis, we have four sperm cells, but again, through meiosis 1 and 2, there's some genetic variation that occurred, right? Synapsis, crossing over, independent assortment. So we still have 4 cells here in oogenesis, but really only one egg. These polar bodies are the other cells that are not functional. And the ovum is only called an ovum after it's been fertilized by sperm. Prior to that, it's called a secondary oocyte. So what's ovulated is a secondary oocyte. But again, similar processes, just the names of the cells are different. This is a nice picture showing the zona pellucida in pink and the dark cells of the corona radiata. Here's just an example of how the sperm has to work its way through those cells, and then that acrosome has that interaction with the zona pellucida. Here we can see here, this one entered, and the nucleus of the sperm combines with the nucleus of the egg to form the, what is that? What is it? The zygote. Yep, the zygote. And we'll pick up in 2 weeks with how that development occurs. So now let's look at your worksheet that I handed out. So number 1, formation of ova and polar bodies is what process? Oogenesis. And the general term for a process that involves meiosis? Gametogenesis. Yep, letter B. The manner in which the zygote is going to divide. So when that zygote is formed, and now it's going to grow and develop, what process is that? Yeah, mitosis. Mitosis. An undifferentiated diploid male reproductive cell. It's diploid, so it's 2N. Yep, spermatogonium. Maturation of spermatid. So we're taking the spermatid and maturing it into a viable moving sperm. Yes, spermiogenesis. And the daughter cell of meiosis 1 in the male. So after meiosis 1, we have Yep, secondary spermatocyte. And the creation of sperm, that whole process from spermatogonium to spermatozoa is spermatogenesis. Yep. So the next question is letter A. What are the two things released in ovulation? What's the one thing I repeated over and over? Secondary oocyte. And what's the partner from meiosis 1 that goes with that secondary oocyte? That small little useless cell? Yeah, the first polar body, we'll call it. And the layer of glycoprotein surrounding the gametes in women and females? No, the corona radiata is zona pellucida. Think of it like a zone. It's like the outer layer that needs to be penetrated for fertilization. Follicle cells surrounding that layer. Corona radiata, yes. So after the sperm penetrates, it completes the second division, resulting in 2 haploid cells. What is that cell called after it's been fertilized? Ovum, and the other cell is, yeah, the second polar body. Number 10 we did already. Number 11 we did already. Number 12, oogenesis, what organ? The ovaries. And what is the structure where the cell is developing and all those cells around it? What are all those cells around it collectively called? Follicle. Yeah, follicle. Where, in what structure does meiosis 2 occur? This is tricky now. Meiosis 2 occurs, it finishes, it completes after fertilization, right? So this is asking, where does fertilization occur? Yeah, in the fallopian tube or uterine tube. And number 13, that solid structure that those follicle cells form is called a corpus luteum, which secretes progesterone. And that concludes lab. The lab exam